Assalamu alaikum. I hope you all are doing well. My name is Dr. Nurilan. So let me share a bit about myself. I'm a doctor and my education journey began at Esteem Army Medical College. And wearing an army uniform was not just an honor, but it was an expression that fueled my passion for serving others. I have registered for the medical exam of the United Kingdom and I'm looking forward to continue my practice in England. I also have a YouTube channel which is dedicated to make medical concept understandable and fascinating. So this was some few things about me. So let's talk about you now. I want to extend a very warm welcome to each one of you. Your contribution and your questions and insights are welcome and important for our learning experience. So let's get started with our first lecture. We are going to talk about physiology, and in physiology we are going to discuss general physiology. Dr. Naman was already teaching you general physiology, and these are the topic which you guys have already studied or covered with him. So if you guys need revision of any topic, kindly drop a comment below in the video description so I come to know that which topics need more revision, as all topics are important from exam point of view. I'm a big fan of reading and learning from the main textbook, and the original book for physiology is Guyton. We have studied Guyton in our MBBS syllabus, and I would recommend that you guys use this book to understand the basic concepts of physiology. In this lecture, we are going to talk about the transport across the cell membrane. This slide is particularly very important. That is the chemical composition of the intracellular and extracellular fluid. And in this slide, it's important that you remember the ions which are important in the extracellular fluid and the intracellular fluid. The concentration of the ions are important because some of the ions are more, the concentration is higher in extracellular and some are intracellular. The important ions which you need to know is sodium and potassium, glucose and amino acids, and proteins, and you must also know the pH. The next important thing is this diagram which shows the transport mechanism. So this diagram is about the lipid bilayer, which is a membrane, the structure of the cell membrane. And the cell membrane is a lipid bilayer. As you can see, they are the lipids that are lipids along with the proteins that are present in the cell membrane. So they are channel proteins, and the other protein is a carrier protein. The channel protein has pores, so the pores, that is basically this opening, whereas the carrier proteins do not have any such pores. And there are three mechanisms that are labeled here: simple diffusion, facility diffusion, and the active transport. So the difference between the diffusion and the active transport is the energy, and the facility diffusion and the active transport both are carried out by the carrier proteins. However, energy is used in active transport. And simple diffusion occurs through the lipid bilayer as well as the channel protein, whereas facility diffusion only occurs through the carrier protein. So this was important. This diagram is important. You must know this diagram, and this needs to be discussed again and again in the further lectures and in similar lecture as well. So what is diffusion? Diffusion basically is the movement of the substances from the higher concentration to the lower concentration. It does not require any energy. There are two types of diffusion. One is a simple diffusion and the other one is a facility diffusion. The simple diffusion can occur through a cell membrane by two pathways. That is directly through the lipid bilayer if the lipid substance is lipid soluble, and the other one is the watery channels. That is, we have already discussed, that are channel proteins. So if we talk about the facility diffusion requires interaction of a carrier protein, and the carrier protein aids passage of the molecules to the membrane and binding chemically with them. This is the same diagram which we have already talked about. Then an important factor to determine how rapidly a substance diffuses through a lipid bilayer is the lipid solubility of the substance. The more the lipid solubility of the substance, the faster they are going to diffuse. And there are some important substances: oxygen, nitrogen, carbon dioxide, alcohols. They are very highly lipid soluble, and all these substances can dissolve directly in the lipid bilayer. The rate of diffusion of each of these substances to the membrane is directly proportional to its lipid solubility. Even though water is highly insoluble in the membrane lipids, it readily passes through channels in protein molecules that penetrate all the way to the membrane. Many of the body cell membrane contain pores that are known as called aquaporins that selectively permit the rapid passage of water to the membrane. The aquaporins are highly specialized, and there are 13 different types in various cells of mammals. So those particles that cannot pass directly to the lipid bilayer, that passes through the channel proteins, and these channel proteins are also known as aquaporins because they selectively permit the passage of water, and these aquaporins are also highly specialized. The next thing is that the other lipid insoluble molecule can pass through the protein pore channel in the same way as water molecule if they are water soluble and small enough. So along with the water, there are other particles that can also pass through the protein through these channel proteins because if they are water soluble and if they are small enough. However, as they become larger, their penetration fall off rapidly. For example, urea. So urea is a larger molecule. So if the size of the molecule increases, their penetration is still going to happen, but the speed of penetration is going to be low. So this is about, so you are going to, what is this in this figure? This is a channel protein or a carrier protein. So as we can see, this is a pore. So it's a gated channels, they are channel proteins. There are two important characteristics of channel proteins. They are selectively permeable and they also have gates. The selectively permeable that they allow this allow permeability. They are permeable to only selective substance. For instance, if there is a sodium ion, then it only allows the passage of sodium ion. If it is a potassium channel, then it only allows the passage of the potassium channel. Other important thing about the channel protein is that they can be opened or closed by gates, and these gates can be regulated by a chemical signal or electrical signals. The next thing is, this is an example of the voltage gated channel, and the sodium potassium channel is a voltage gated channel. And these channels are very important to know because they are very important how the nerve impulses are conducted in our body and how the muscles contract. So that is why these channels, it's important that you must know about these channels because they are going to be a damage in diseases which we are going to discuss in pathology. So that's very important that you know the basic physiology or the basic normal human mechanisms and how the ions are conducted and how the ions and substances are transported in the cells, so that you know these mechanisms of impulse conduction and the muscle contraction. So facile diffusion, we have already talked about. It occurs through the active transport. It occurs through the carrier proteins. Glucose and amino acids, they are the example of the facile diffusion. So in the case of glucose, at least 14 members of the family of membrane. So the example of the transport of glucose consists of the group 4. Group 4 is an important glucose transporter. It is activated by insulin, which can increase the rate of facile diffusion of glucose as much as 10 to 40-fold in insulin sensitive tissues. Insulin sensitive tissues include the adipose tissue and the skeletal muscle. The factors that affect the net rate of diffusion is the concentration gradient, membrane electric potential, and the pressure difference. The concentration gradient is the net diffusion, that is the outside and the inside. If there are more particles outside, the difference is more important. That if the difference is more, then the net rate of diffusion is going to be more. Moreover, we see the membrane electrical potential. Then if there are more negative charge outside the cell and we have introduced a negative charge, a positive charge inside the cell, then negative and negative will move towards the positive as opposite attracts. So there will be a movement from the left to right. If you see the factors that affect the net rate of diffusion, the other factor is the pressure difference. Pressure difference is very important in the capillaries. There is a movement of the blood across the capillaries because of the pressure difference from higher pressure to lower pressure. Now we are going to talk about the osmosis. Osmosis is simple diffusion of water, or it could be simple in the water movement, and the water moves from the higher concentration to the lower concentration through the semi-permeable membrane. The osmotic pressure is also very important. The definition of the osmotic pressure is the amount of pressure is required to stop osmosis, and it is exerted by particle in a solution. Whether their molecular ion is determined. So it's important to know that osmotic pressure is determined by the number of particles per unit volume, not by the mass of the particles. It's important to know the concept of osmole. Osmole is the number of particles. For example, there are two particles, glucose and sodium chloride. So if you put glucose in water, it won't dissociate. So glucose has just one osmole, where sodium chloride will dissociate. So sodium chloride has two osmoles. Osmolality is a normal osmolality of the extracellular and intracellular fluid is about 300 milliosmol per kg of water. So the normal osmolality value, you must know this because it is the range in number of diseases. Now we're going to talk about the active transport. So that's two important things that you need to remember for active transport. The one, it is a mechanism that involves energy and always occur against the concentration gradient and involve the carrier proteins. So there are two types. Active transport, when cell membrane moves the molecular ions uphill against a concentration gradient, or an electrical gradient, or a pressure gradient. This is known as the active transport. In both instances, transport depends on the carrier protein that penetrates to the cell membrane. However, in active transport, the carrier protein functions differently from the carrier in facile diffusion because it has the capability to impart energy to transport substance to move it against the electrochemical gradient. So there are two types of active transport, primary active transport and secondary active transport, and they are being primary active transport and secondary active transport, and they are basically divided according to the source of energy used to facilitate the transport. Primary active transport, the example of the primary active transport is a sodium potassium ATPase pump, which is very important in conduction of impulses. This is a very important transporter that is present. 3 sodium ions move out of the cell and 2 potassium ions move into the cell, and ATPase is going to catalyze ATP into ADP. Energy is used in order to move the ions against the concentration gradient. Now we're going to talk about the sodium potassium pump is also important for controlling the cell volume. Unless the process of sodium potassium is checked, the cell will swell indefinitely until it bursts. This is a normal mechanism for preventing this outcome of the sodium potassium pump. The other thing is electrogenic nature of the sodium potassium pump. Sodium potassium pump is very important because of the electrogenic nature. It creates an electric potential across the cell membrane, and it's a very important concept because it allows the transmission of impulses and the muscles contraction. So there are 2 other more examples of primary active transport. One is the calcium and the other is the hydrogen ion. It's important for you to remember the active transport of the hydrogen ion in the stomach and in the kidneys, because in the stomach HCl is present, as we all know, the gastric acid. So hydrogen ion transport is very important in the stomach, and hydrogen ion transport is also very important in the kidneys. The next important thing is the co-transport and the counter-transport. So secondary active transport is the energy is dissipated, but it's not directly used by the same ion. It's involved in the movement of the other ion. If the direction of the movement is in the same direction, then that is known as a co-transport. And if the movement of the ions are in the opposite direction, then that is known as a counter-transport. So when sodium ion transported out of the cell by primary active transport, a large concentration gradient across cell membrane usually develop, and low concentration, this gradient represents the storage of energy, because excess sodium outside the cell membrane is always attempting to diffuse to the interior. Under appropriate condition, sodium can pull the substances along with the sodium through the cell membrane. This phenomenon is known as co-transport, as a form of the secondary active transport. In counter-transport, the substance to be transported is on the inside of the cell and must be transported to the outside. So that is the opposite transport. So sodium glucose co-transporters are important in transporting glucose across the renal and the intestinal epithelial cells, and sodium counter-transport of the calcium and hydrogen ions are the example of the counter-active transport. Another important thing is the cellular sheets. So in some tissues, the cells are arranged in cellular sheets, and there are different surfaces. There is a brush border, there is a basement membrane, then there is also the lateral surface, and there are various mechanisms that are involved in the movement of these ions. So in the movement of these ions, for instance, at the brush border, the mechanism involved is the diffusion. Sodium is diffused into the cell, whereas at the lateral border, the active transport is involved, and as well as the basement membrane, the active transport and osmosis is involved. So this was all about the how substances are moved across the cell membrane. This is very important topic. You must know this in order to understand the other concept of physiology. These concepts form the basis of the physiology. So this will all. Thank you for listening, and please subscribe my channel, and I will definitely share all my physiology lectures over this channel, and I will make a playlist especially for you guys. And if you guys need any help in any other medical concept, or if you want me to make videos on any other topic which you are unable to understand, it could be topic of anatomy, or it could be topic of community medicine, or any other subject, or even pharmacology. So I will definitely help you guys to understand the concept and make it easier for you all. So thank you. If you have any query, you can drop a comment. So that's all.